Okay, happy Friday. I am shooting this a week before you're supposed to see it, but today's Friday too. You're supposed to get this on Friday of next week, so hopefully you've had a good week and it's a good Friday for you. Hopefully you're looking forward to the weekend, and I have no idea what's going on next weekend because we still don't have football, but that's okay. Today we're going to talk about enzymes, and hopefully you're in comfy clothes and you can just kind of sit back and take this in. I hope that I get a chance to hear from some of you. I've heard from a lot of you. That's awesome. That's great. I've given out my Google Voice text number that you can text me at. You can also email me anytime. I try to keep up to date with all of it to help you kind of figure out this online learning. So if you haven't talked to me and you want to shoot me a quick hello sometime, I would love it. I really want to get to know you. I can't wait to meet every single one of you. Today we're talking about enzymes. So enzymes, what we need to know, or you as students need to know, our TEK is 9C. You need to be able to identify and investigate the role of enzymes. And so that's what we're talking about today. So I'm going to move my screen in just a minute as soon as it loads. And hopefully my video part won't be in the way. But that's what we're going to do today. And let's see. Okay, so what are enzymes? See, I just moved myself and then mentioned it. Okay, what are enzymes? Enzymes are proteins that help molecules react with one another. Okay, so they're proteins. That's really important. Enzymes are proteins. That's one of the main functions of proteins. Okay, they're monomer or amino acids because they're proteins. Okay, and then enzymes are used by cells to trigger and control chemical reactions. So they're used by cells to kind of speed things up. Okay, without enzymes, several reactions in cells would never occur, or they would happen so slowly that it wouldn't be useful. Okay, and in digestive enzymes, it often ends with an ACE. Okay, all right. So let's talk about what a chemical reaction is. You learned this one in sixth grade. I know I taught my sixth graders this. Chemical reactions are when two things are put together and they create a totally new substance. Okay, so at the very base understanding of what a chemical reaction is when two things are put together, such as hydrogen and oxygen, which are gases, are put together and they make water. That's a chemical reaction. Okay, so when hydrogen, the element, and oxygen, the element, are put together, it makes something new called water. When you have a fire, okay, you strike a match on phosphorus and it becomes a campfire. That campfire is a chemical reaction. It becomes something totally new that usually cannot be reversed. And you've talked about signs of chemical reactions in earlier grades, but a rotting banana. You had a banana, it sat out in the oxygen, it can't be reversed. You can't have a new banana again. You have to put it in the refrigerator for a smoothie or something, okay? So a chemical reaction is the process of changing one set of reactants, so that was the hydrogen and the oxygen, into another set called products. And by rearranging the atoms, it becomes something totally new. Reactant bonds are broken and new ones are formed to form a new product. So baking soda and vinegar. You have baking soda, you have vinegar, and you put them together and they form this cool thing that you can put in science fair volcanoes. I don't know, but it's bubbling. It becomes something new. So the baking soda and the vinegar were reactants, and then your product was the bubbling thing that becomes a lot of science fair stuff. All right. Oh. So then let's talk about enzymes. So how do they affect chemical reactions? So how do they work? You have these parts of enzymes, okay? The substrate or the reactant moves toward the enzyme's active site. Okay, so the purple is the enzyme, okay? And this is the active site right here, which we're going to talk about later. And then the substrate is the red part. So it is moving toward the enzyme's active site. The chemical reaction is triggered by the enzyme. So you have something is triggered by this enzyme because it's there, and then the enzyme releases the product. Okay, and so that's why it became a different color. So you have a substrate coming into an enzyme at the active site, it attaches to it, and then when it goes into a totally different product. That's how they work. That's kind of the function of them. So let's talk about a few keywords that have to do with enzymes. First of all, an enzyme is a catalyst. A catalyst. A catalyst is a substance that speeds up the rate of a chemical reaction, so it makes it faster. It lowers the energy needed to do the reaction, so it lowers what's called the activation energy. So it's not the reaction, it's not a product, it's not a reactant, it's something different. It's something different that comes into this reaction that's already happening. It's like a helper, if you want to say it like that. It participates in the reaction, but it doesn't change itself. Therefore, it can be used over and over again. So you can reuse enzymes. That's important. Enzymes can be reused over and over again. So the illustration on this is that the dad is the enzyme. He's a helper. He's helping the little boy learn how to hit the ball faster. Because if the boy just stood there and he's like, "What do I do?" The boy would eventually hit the ball. He'd figure out what to do. But if the dad or the coach is telling the boy how to hit the ball, then he is a catalyst, or he is helping the reaction. So the dad is like an enzyme, or the coach. Either way. Okay, so what is a substrate? So here's another word that you need to know. A substrate, enzymes bind to or break molecules called substrates. So here's your enzyme in the red, and this is the substrate, which is coming into the active site. So this was part of the reactant, okay? So it's coming into the active site. These substrates are the reactants that are catalyzed by the enzyme. So the substrate is what's attaching to that enzyme in order to become something new. The active site, I'm just having to move myself all around, sorry. The active site. So the active site I'm pointing on my screen like you can see it. The active site is that area that the substrate is going to attach to. It's called the active site. Each substrate and enzyme has a specific shape. Therefore, enzymes bind to substrates based on the shape. So it's kind of like the amoeba sisters will talk about it, but like Pac-Man has that triangle shape. They have specific shapes in enzymes so that certain substrates fit like a puzzle piece into it. The site on the enzyme where the substrates bind are called the active site. So you can see that on this one, and you'll see that kind of as we move along. So the product, let me move myself again. The product is what you're going to have at the end. You probably knew that already. The enzyme converts the substrates. Those are the red and yellow here on this one, in this illustration. The enzymes are going to convert the substrates into totally different molecules called products. It's a chemical reaction, remember? So it becomes something totally new. That's why it's orange. So we have red and yellow and makes orange, yay. The product is what is released at the end of the reaction. All right, so there's a couple different ways that enzymes work. One of the theories is called a lock and key hypothesis, and it is kind of like how it sounds. So enzymes bind to the substrates based on their shape, so the complementary shape. The fit is so exact that the active site and the substrates are compared to a lock and key, kind of like what I put over here in the illustration. Most digestive enzymes are named for foods that they help. So lactase helps break down lactose or milk and sugar. Okay, so don't get too caught up in the names right now. I'm going to give you a vocabulary sheet for prefixes and suffixes you'll need to know later. But what you need to know is just that there's a complementary shape on an enzyme for the substrate to fit into. What are other examples of enzymes and what they break down? You have lactase, which breaks down lactose and glucose and galactose. Big words, I know. We're just going to go slow with this. You have sucrase, the sugar clipper. It breaks down sucrose into glucose and fructose. You have amylase, breaks down starch in your mouth and stomach. Lipase, which breaks down fats. and pepsin, which breaks down proteins. You don't necessarily have to memorize this part. Like, you don't have to memorize all these detailed enzymes and things like that. That's not going to be on your test. If they're there, it'll have what it means. What you need to know is there's lots of different enzymes with lots of different functions. How are enzymes affected by a reaction? Enzymes are not changed by the reaction they catalyze. They're reusable. So I want you to know that enzymes don't die. Well, they're not going to die anyway because they're not living. But enzymes don't just go away and get destroyed completely. They're reusable if they're a good enzyme. Okay? How can enzymes be affected? Enzymes each work best at a specific temperature and pH, and we're going to talk about that a little bit more. Temperatures outside the correct range can cause enzymes to break down or change shape, and the breakdown is called denaturization. And that word's going to be important because you're going to see that on a quiz and test next week. So I'm going to talk about it on another slide. But denaturization is when an enzyme is destroyed because of the environment, the effects. It's affected by the environment. So here's a little bit more about denaturization or it being destroyed. When enzymes in a reaction are denatured, it means that they no longer work to continue the reaction. So they're not, or they slow a reaction down because now it takes more energy. So what does that mean for you? So it's kind of like a stoplight. You know, when the stoplight's working, traffic builds up and traffic goes just like it's supposed to. Everybody follows the stoplight. But let's say that stoplight, you know, on a long road, if the stoplight's working, we'll all just stop, go, you know, yay, we'll all get there on time, or we'll stop so the other cross people can go across. But let's say that stoplight stops working. Everything past the stoplight doesn't work anymore. So that means you have a big buildup of traffic, a big buildup of cars. So that means all of the stuff that's behind it starts increasing again. So there's a big buildup. So you need the enzyme, like the stoplight, to continue working like it's supposed to so that traffic can continue like it's supposed to. What that means is that the rate of the reaction will slow back down, causing the substances the enzymes were being used for to increase right before it stops. So see how all that traffic got a whole bunch, like there's all these, hopefully it's not a wreck. There's all these traffic buildup right behind that stoplight because now it's a stop and go light. It never turns green. The stoplight's broken. It's irritating. You know, everybody's slowed down. All of these cars increase right before it got denatured or destroyed the stoplight. Okay? Because it can't move on to form the new product without a lot more energy, everything just kind of halts. Okay? And whatever is right before it, whatever reactant is right before it will increase. Hopefully that makes sense. I wish y'all were in my classroom with me. Okay. This happens when the enzyme is exposed to extreme like pH or high temperatures or like really high salinity in the ocean, which is salt, or alkalinity, which is acidic. So anytime the environment is not right for the enzyme or in that certain range, it'll get denatured or destroyed. Okay. One of the other things I wanted to talk about today, because you're going to see this on quizzes and tests, if I can move my camera, is how to use and how to read enzyme graphs and enzyme action. Okay, so this one right here is showing you the amount of energy supplied and released when you have an enzyme and when you don't have an enzyme. So reactions still happen, like life still goes on, okay, with or without an enzyme. But the enzyme helps it to speed it up. The enzyme means that you need less energy to make the reaction happen. That's speed it up is what catalyst means. So if you look at this graph right here, you have to be able to read graphs. What's happening on the red line, if you follow along with me, is that there's no enzyme attached to it. So if I put hydrogen and oxygen together, this is not necessarily where an enzyme is used, but I'm just thinking of a simple reaction that you guys know. If I put hydrogen and oxygen together, it's still going to make water. Okay, and it's going to take this red line amount of energy, this high amount of energy to make it happen. But let's say I attached the correct enzyme to it, which again, you don't usually use enzymes with hydrogen and oxygen. But let's say I'm going to attach an enzyme to it. Well, now, if I attach an enzyme to it, it doesn't take as much energy. It doesn't take as much energy for that to happen. So it happens sometimes quicker. It happens at a lower amount of energy. And the activation energy needed is much less with the enzyme than without it. Okay, so that's how you read this graph. It's really important when you're reading graphs and charts and things like that, that you look at what's on the y-axis, which is the up and down axis, and you look at what's on the x-axis. Everything in this graph is a clue, and so everything tells you a story or gives you a hint about what's happening. And a lot of times in science, the answers are actually just in the hints or in the clues that are all around the picture. Just letting you know. Okay, here's another graph that you're going to see. So if you look on the y-axis, it says the rate of the enzyme activity. So that's the rate at which it's happening. So the speed and rate are the same thing. So as I go up the y-axis, it's going faster, it's increasing. Okay, the x-axis is temperature. So now what we're reading is what is the optimal temperature here for the enzyme to work? And this says what happens to enzymes at low temperatures? They don't work. What happens when they're denatured? They go back down, because once it gets to a higher temperature, it'll go back down. But the optimal, optimal means best. The best temperature is going to be right here. And there's no numbers here. I wish there were, but there's not. I forgot to get one with the numbers. But you're going to look at the highest top of the hill when it's working at its best. That's the best speed. That means that's the best temperature, okay? And so that's how you read that line graph. And then another graph you might see is one that talks about pH. So what is the acidity? What do you need for this enzyme to happen? So the y-axis is the enzyme activity. So you have a low enzyme activity. It's sort of not working at total speed. And then you speed it up. There's more activity. And then here's pH. You have bases along this line and acids down here. I'm not sure if you've learned this yet in 7th and 8th grade. But when you're reading this chart, there's two different enzymes being charted. There's an enzyme A, which is blue, and enzyme B, which is red. Okay, so the optimal pH for A, which is blue, would be a pH of around 6. My line's not very straight when I put it in there. We'll just say around 6. The optimal pH for B is going to be around 10. You see what I'm saying? You're going to see charts like this on quizzes coming up next week and your test coming up next week. You have a test next Friday on biochemistry, scientific method, and lab safety, and I want you to make sure that you know what's going on in these slides. Okay, finally, why are enzymes called the body's workers? Nearly every reaction in your body is helped by an enzyme. The biggest thing I want you to remember is that enzymes are proteins. Enzymes are proteins. So that's really important. Hopefully this makes sense. If you get stuck or something doesn't make sense, please ask me. You have lots of different ways to communicate with me. Please ask me. I'm here to help. All right, I'll see you next week.